Welcome friends to a new session on molecular biology. In this session, we will deal with organization of eukaryotic and prokaryotic chromosomes. So far, we have been dealing with DNA, RNA, but don't you think that if they are in highly organized inside the cell or nucleus, cell have been translating, transcribing or replicating throughout its life meaninglessly? This mystery dissolved by the discovery of chromosomes. Cell wraps the whole genome well and good, protected and make available for them if and only if the situation demands. Discovery of chromosome. The word chromosome means colored bodies. It contains the cell's genetic material which determines how an organism develops. Although Mendel provided convincing evidence that inherited traits were governed by discrete factors or genes, but his studies were totally unconcerned about the physical nature of these elements or their location within the organism. With the discovery of DNA in 1953, one of the greatest breakthroughs made it possible to express the biological concepts and structures in concrete chemical terms. The rediscovery of Mendel's work and its confirmation had an immediate influence on research in cell biology. Whatever be their physical nature, they have to be inherited in a consistent manner with Mendelian principle. The term chromosome was first suggested in 1888 by a German anatomist, Heinrich Wilhelm Gottfried von Waldner-Hartz, 1936 to 1921. He used the term to describe certain structures that form during reproduction. In 1903, Walter Sutton, a graduate student at Columbia University, published a paper that pointed directly to chromosomes as the physical carriers of the Mendel's genetic factors. Sutton followed the development of sperm cells in the grasshopper because they got large and easily observable chromosomes. On examination of spermatogonia, Sutton counted 23 chromosomes and suggested that they are present in look-alike pairs. Sutton soon realized that they are organized in pairs or called them as homologous chromosomes and correlated perfectly with the pairs of inheritable factors uncovered by Mendel. What are chromosomes? Chromosome is a single piece of coiled DNA containing many genes, regulatory elements and other nucleotide sequences. The word chromosome comes from the Greek word chroma, means color, and soma means body. During interphase, when cells are not dividing, the genetic material exists as a nucleoprotein complex called chromatin, which is dispersed through much of the nucleus. Further folding of the chromosomes occurs in the mitosis, producing visible metaphase chromosomes. Now, chromosome morphology. Early microscopic observations on the number and size of chromosomes and their staining patterns led to the discovery of many important general characteristics of chromosome structure. Each metaphase chromosomes consist of two sister chromatids, which are attached to a centromere. The location of the centromere on each chromosome gives a chromosome its characteristic shape and can be used to help describe the location of specific genes. This figure shows the schematic representation of chromosome inside the cell. Each chromosome has one centromere with one or two arms projecting from the centromere and a telomere. A centromere is visible as a primary constriction on mammalian chromosomes. In humans, this site is characterized by the presence of a particular type of tandemly repeated symbol DNA sequence called alpha satellite. Alpha satellite DNA is a 170 base pair sequence repeated over many millions of base pairs of DNA. Telomeres are specialized structures at the ends of chromosomes. Telomeres hide the ends of the chromosome from the mechanism within the cell that monitor DNA damage. They are also needed to overcome the problem of end replication. Chromosomes are classified according to the centromere position. First one, metacentric. Metacentric chromosomes are V-shaped, and in these chromosomes, the centromere divide roughly into two equal arms. In the case of amphibians, it have metacentric chromosomes. Second, submetacentric chromosomes. In submetacentric chromosomes, centromere is submedian, giving one longer and one shorter arm. The submetacentric chromosomes are L or J shaped during anaphase. Third one, acrocentric. In acrocentric chromosome, the centromere is located terminally and forms two unequal arm lengths. The locust acrididae have the acrocentric chromosomes. The P arm is too short to observe and they appear rod shaped during anaphase. Please see the figure on screen. It shows the morphology of chromosome according to the position of centromere. Fourth one, telocentric. In telocentric chromosome, the centromere is located at one end. They appear rod shaped during anaphase. Now, the cell cycle. The diploid cells of our body are derived from the original diploid fertilized egg by repeated rounds of mitotic cell division. Each round can be summarized as one turn of the cell cycle. This comprises a short stage of cell division, the M phase, that is mitosis, and a long intervening interphase. Interphase can be divided into S phase, DNA synthesis, G1 phase, gap between M phase and S phase, and G2 phase, it is a gap between S phase and M phase. From anaphase of mitosis right through until DNA duplication in S phase, a chromosome of a diploid cell contains a single DNA double helix and the total DNA content is 2C. G1 is a normal state of a cell and the long term end state of non-dividing, that is spacing cells. Cells enter S phase only if they are committed to mitosis. Non-dividing, that is spacing cells, remain in a modified G1 stage, sometimes called as G0. Please see the figure on screen. It gives the different stages in cell cycle. Interphase chromatin. During interphase, that is the period of the cell cycle where the cell is not dividing, two types of chromatin can be distinguished. The density of the chromatin that makes up each chromosome, that is how tightly it is packed, varies along the length of the chromosome. During interphase, most of the chromatin, that is euchromatin, exists in an extended state, dispersed through the nucleus and staining diffusely. While staining, heterochromatin stains denser than euchromatin. Please see the figure on screen. It describes the chromatin condensation changes during the cell cycle. Euchromatin. Genes located in this region may or may not be expressed depending on the cell type and its metabolic requirement. Heterochromatin, either due to naturally or chromosomal rearrangements, are very unlikely to be expressed during these chromosomal stages. Heterochromatin can be further distinguished into two types. Constitutive heterochromatin. This is always inactive and condensed. It largely consists of repetitive DNA and is found in and around centromere. Facultative heterochromatin. It can exist in either genetically active, that is decondensed, or inactive, that is condensed form, as in the case of mammalian X chromosome inactivation. This figure shows the euchromatin and heterochromatin. Individual chromosomes cannot be distinguished at this stage. They appear in nucleus as a homogeneous tangled mix of DNA and protein. Now, special types of chromosomes. Eukaryotes possess some unusual types of chromosomes in some body cells or at some particular stage of their life cycle. And the special types of chromosomes include polytene chromosomes and Lamprecht chromosomes. Now, polytene chromosomes. Polytene chromosomes are gigantic interphase chromosomes that provide an important model for the analysis of the genetic organization of chromosomes and genomes as a whole. Polytene chromosomes develop from the chromosomes of diploid nuclei by successive duplication of each chromosome element, that is chromatid. They have been found in many tissues of the representatives of two orders of insects, Diptera and Columbola, in the macronuclear analgon of Infusoris, in certain organs and tissues of mammals, and also in the cells of synergids, antipods, and endosperm of angiospermous plants. Polytene chromosomes were discovered by Balbani in 1881 and named by Koller. He observed unusually long and wide chromosomes about 100 to 200 times in size of the chromosomes in meiosis or mitosis in the nuclei of the salivary gland cells of the larvae of dipterians like Drosophila. This is particularly surprising since the salivary gland cells do not divide after the glands are formed, yet their chromosomes replicate several times, a process called endomitosis, and become exceptionally giant-sized to be called polytene or multi-stranded chromosomes. The endomitosis process results in the production of two X chromosomes, where X gives the number of multiplication cycle. Firstly, the formation of polytene chromosome is associated with the elimination of the entire mechanism of mitosis after each DNA doubling, as a result of which the cell cycle consists of just two stages, that is synthetic S and intersynthetic, that is G. Secondly, at the end of each replication period, DNA strands do not segregate. Rather, they remain paired to each other to different degrees. Thirdly, the polytene chromosomes formed are incapable of being involved in mitosis. Fourthly, the nuclear membrane and nucleolus remain intact during consecutive DNA replication cycles. The morphology of polytene chromosome can vary widely due to the differing degrees of synapses of the DNA strands. Each strand within polytene chromosome will be conditionally designated as chromatid. If homologous chromatid conjugation is maximal, classic polytene chromosomes, that is cylindrical cables with a distinct banding pattern such as those found in Chironomus tendens or D. melanogaster, were formed. However, the degree of chromatid conjugation is minimal. A polyploid nucleus with a reticular structure, namely cryptic polyteny, is formed. Please see the figure on screen. It shows the polytene chromosomes. Lambrish chromosomes. Lambrish chromosomes were discovered in salamander egg cells, Amplystoma mexicanum, by Fleming in 1882. These chromosomes occur in the oocytes, that is germ cells in the ovary of amphibians and in some insects. They are extremely large synapsed homologous chromosomes which can be seen in the diploidine stage of prophase 1 in meiosis. They measure about 1500 to 2000 micrometer in length. A Lambrish chromosome consists of an axis from which paired loops extend in opposite directions, giving the appearance of a Lambrish. The axis consists of chromomeres, that is nucleosomes, and intrachromomere regions. The loops consist of transcriptionally active DNA which can synthesize large amount of mRNA necessary for the synthesis of yolk. Please see the figure on screen. It shows the Lambrish chromosome. A routine analysis of Lambrish chromosomes can only provide detailed description of their morphology. Transcriptional activity of Lambrish chromosomes can be even observed under a light microscope and can be determined for morphological changes. Changes in transcriptional activity might be a scale for assessing the activeness of the chromosome. More loops means transcriptionally active and shrinking of the side loops denotes a decrease in the activity. In transcriptionally active regions of chromosomes, histone proteins give way to non-histone proteins, inducing the loss of nucleosomal structure of the coding chromosome segments that assume the shape of a loop. The average length of a typical Lambrish chromosome loop is 10 to 15 micrometer, though some can be as long as 50 or even 100 micrometer. The loops can be classified according to the type of the transcriptional polymerase. The largest loops include those transcribed by polymerase II. The smallest loops are transcribed by polymerase III. Now, the organization of chromosomes. All organisms are derived from cell division and they are derived from other cells. Prokaryotes lack a defined nucleus and have a relatively simple internal organization. The genome of prokaryote in particular consists of a single small circular chromosome in which DNA is not packaged in any obviously organized way. In the case of eukaryotes, intracellular organization is much more compact with well organized cytoskeleton and an intact nucleus. They got linear chromosomes in their cell nuclei, in each of which a single very long DNA molecule is elaborately packaged by histone and other proteins. Now, the chromosome size. The genome size in eukaryotes varies widely. The size of eukaryotic genome considered as 10 to 1000 MB. The size of viral genome also is 10 to 1000 MB and the bacterial genome seems to be between 1 to 10 MB. Different organisms have different numbers of chromosomes. For example, humans have 23 pairs of chromosomes, 22 pairs of number chromosomes called autosomes and 1 pair of sex chromosome called X and Y. Each parent contributes 1 chromosome to each pair so that offspring get half of their chromosomes from their mother and half from their father. In eukaryotes, they are condensed to form a structure called chromatin so that they can be packed to fit the cell nucleus. The structure of the chromosomes and chromatin vary within the cell cycle. The haploid human genome contains approximately 3 billion base pairs of DNA packaged into 23 chromosomes. During mitosis and meiosis, various degrees of compaction takes place that results in the classic 4-arm structure with centromeres in the center and sister chromatids attached to it. Any aberrations or manipulation during its inheritance or duplication causes catastrophic impact, even leading to cancer. Now, differences between eukaryotic and prokaryotic chromosomes. In prokaryotes, it got single circular chromosome. In eukaryote, multiple linear chromosomes are there. In prokaryotes, it contains only single copy of each gene, that is, they are haploid. But in the case of eukaryote, it contains 2 copies of each gene, that is, they are diploid. In prokaryotes, it is made only of DNA. In eukaryotes, it is made of chromatin, a nucleoprotein, that is DNA coiled around histone proteins. In the case of prokaryotic DNA, it can interact with the cytoplasm, transcription and translation occur simultaneously. But in the case of eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. In prokaryotic genomes, they are efficient and compact, containing little repetitive DNA. In the case of eukaryotes, contains large amounts of non-coding and repetitive DNA. Now, packaging of chromosome, it requires multiple hierarchies. The basic repeating unit of chromatin is the nucleosome. The packaging of DNA into nucleosomes shortens the fiber length about 7-fold. In other words, a piece of DNA that is 1 meter long will become a string of beads chromatin fiber just 14 centimeters, that is about 6 inches long. Despite this shortening, is still too long to fit into the nucleus. In the cell, the structure of each chromosome is highly ordered. Even in the interphase nucleus, the 2 nanometer DNA double helix is subject to at least 2 levels of coiling. Nucleosome. This consists of a central core of 8 histone proteins, small, highly conserved basic proteins of 102 to 135 amino acids. Each core comprises 2 molecules, each of histones, that is H2A, H2B, H3 and H4, around which a stretch of 146 base pair of double stranded DNA is coiled in 1.75 turns. Adjacent nucleosomes are connected by a short length of spacer DNA. Electron micrographs of suitable preparations show a string of beads appearance. The string of beads, approximately 10 nanometer in diameter, is in turn coiled into a chromatin fiber of 30 nanometer diameter. The interphase chromosome seems to consist of these chromatin fibers, probably organized into long loops. Please see the figure on screen. It shows the hierarchical level packaging of chromosome. Now, chromosome organization. There are several levels of organization: solenoid, hollow contact helix of nucleosomes, chromatin fiber, scaffold, and finally chromosome. First, packaging is done to wrap 2 nanometer diameter duplex DNA to obtain a nucleosome fiber with 11 nanometer diameter. This is again condensed to form a 30 nanometer chromatin fiber. Loops of the 30 nanometer chromatin fiber containing 20 to 100 kb of the DNA per loop are attached to a central scaffold. Finally, in the chromatids of a metaphase chromosome, the loop scaffold complex is compacted yet further by coiling. In higher levels of chromatin organization occurs as solenoid model, in which 30 nanometer fiber exhibits a regular helical organization consisting of 6 to 8 nucleosomes per turn. Core histones of adjacent nucleosomes may interact with one another by means of their long flexible tails. Sterease indicates that N-terminal tail of an H4 histone from one nucleosome particles and the H2A, H2B dimer of the adjacent particles. These interactions are thought to mediate the folding of the nucleosomal filament into a thicker fiber. Now, histones. Histones are remarkable small proteins that possess an unusually high content of the basic amino acids, mainly arginine and lysine. They orderly package eukaryotic DNA and protect DNA from nuclease digestion and other types of information. There are 5 types of histones on the basis of arginine-lysine ratio. These are basic amino acids. They are H1, H2A, H2B, H3 and H4. The histone core of each nucleosome consists of 2 copies of each histones, that is H2A, H2B, H3 and H4. The remaining one, that is H1, resides outside the nucleosome core particle and is referred as linker histone because it connects part of the linker DNA that connects one nucleosome core particle to the next. These 5 are arranged as octamer. Together, the H1 protein and the histone octamer interact with about 168 base pair of DNA. The core particle itself consists of approximately 1.8 turns, that is 146 base pair of negatively supercoiled DNA wrapped around 8 core histone molecules, that is 2 each of H2A, H2B, H3 and H4. The linker histone binds an additional 20 or 30 base pairs of DNA. Please see the figure on screen which shows the histone proteins along with the DNA wrap around it. However, the presence of nucleosomes and the folding of chromatin into 30 nanometer fibers pose barriers to the enzymes that unbind and copy DNA. It is therefore important for cells to have means of opening up chromatin fibers or removing histones transiently to permit transcription and replication to proceed. Now, non-histone proteins. The non-histone proteins are mostly acidic and have been considered more important than histone as regulatory molecules. Some non-histone proteins also have enzymatic activities. The most important enzymatic proteins of chromosomes are phosphoproteins, DNA polymerase, RNA polymerase, DPN pyrophosphorylase and nucleoside triphosphatase. The metal ions as calcium and magnesium are supposed to maintain the organization of chromosomes intact. Now, C-value paradox. The C-value is the total number of DNA nucleotide residues in the genome, that is per haploid set of chromosomes. When we compare this to the complexity of the organism, we find a massive disparity. Clearly, the amount of DNA is not proportional to that required to produce all the proteins made by the organism. The failure of C-value to correspond to phylogenetic complexity is called the C-value paradox. The figure shows the C-values of different organisms. This perplexing variation in genome size occurs mainly because eukaryotic chromosomes contain variable amounts of DNA with no demonstrable function, both between genes and within genes in introns. Now, let me summarize the session. Chromosomes are organized in beads on a thread. The packaging is carried out in several stages of packing like folding, scaffold formation and finally to highly condensed chromosomes. The basic repeating units of chromatin are nucleosome. The chromosomes are usually associated with basic proteins called histones and non-histone proteins. Histones are remarkable small proteins that possess an unusually high content of the basic amino acids arginine and lysine. There are 5 types of histones on the basis of arginine-lysine ratio. These are the basic amino acids. They are H1, H2A, H2B, H3 and H4. These 5 are arranged as octamer. Together, the H1 protein and the histone octamer interact with about 168 base pair of DNA. Here are some assignments for you to work out. First one, what are chromosomes? Differentiate between eukaryotic and prokaryotic chromosomes. Second one, define the terms acentric, acrocentric and polycentric. Third one, explain with a neat diagram 4 morphological shapes attained by the chromosomes. Fourth one, differentiate euchromatin and heterochromatin. Fifth one, what are polytene and Lamprecht chromosome? Now, I can suggest some books for your further studies. The Cell, A Molecular Approach, 3rd edition, Cooper MG, 2007, published by ASM Press, Washington DC. Molecular Cell Biology, 4th edition, written by Lodish H, Burke A, Sipersky SL, Matsudaira P, Baltimore D and Darnell, year 2000, published by W. H. Freeman Publishers. Cell and Molecular Biology Concepts and Experiments, 7th edition, written by Karp G, year 2013, Wiley Publication. An Introduction to Genetic Analysis, 7th edition, written by Griffith J. F., Miller J. H., Suzuki D. T., Lewontin C. R. and Gilbert W. M., year 2000, published by W. H. Freeman. Human Molecular Genetics, 2nd edition, written by Strachan T. and Reed A. P., year 1999, publisher Wiley List, New York. You can visit these websites for further information. www.tnau.ac.in/eagri50/GBPR111/LEC13.pdf www.pfb.info.pi/www.pfb.info.pl. Thank you for watching this session. See you next time. Bye.